Hello everyone. So I welcome you back to another important lecture. And for today, we would look on toward one of the major type of the reproduction, which is followed by the living organism, and this goes with the name as asexual reproduction. Whenever the word asexual is coming, so what you have to keep it in your mind as that there would be no involvement of the sex cells, and you know that these sex cells they are the haploid cells. To what we also refer to them as the gametes, and if we were to talk about us, the human beings, so our gamete cells or the sex cells, they are the sperm cells and the egg cells that later fertilize and ultimately makes the zygote, and finally the human being emerges out of it. But in asexual, there would be no such need of the sex cells or the fertilization situation. And in here, there is only involvement of one parent. So that means that there would be no fusion of gametes and there would be no mixing of the genetic information from the two parent. And as a result of it, any of the offsprings that we would be forming, so they all would be genetically identical to the parents, or in other words, we could say that they would be exact clones or the copies of the parents. So that is why, in terms of remembering the definition of the asexual reproduction, so this is a process that results in genetically identical offsprings and the two have come from where? One parent. So if we were to talk about that what organisms are following this form of the reproduction, so we say is that this is followed by the bacteria and this would be initiated by the plants with us. For the bacteria, their asexual reproduction would be termed as a binary fission, and for the plant, the asexual reproduction would go with the name, and that name technically would be the vegetative propagation with us. So if I were to quickly take you to be understanding that how come the process of the asexual reproduction or the binary fission will go in case of the bacteria, so you just have to look on this diagram. Now you see as that at the top I have got this parent bacteria with me, and this parent bacteria at the end has to be making two genetically identical daughter cells or the bacterial cells, and I have to be making sure as that whatsoever the genetic content or the cellular content that this parent bacteria has got, the same must come into their respective daughter cells. So that would mean as that prior to be compelling this parent bacteria to be undergoing the binary fission, so what I am required to do is that any of the cellular content that we have in this case, as if you see, I have got this DNA molecule, I have got these ribosomes, and I have got this extra genetic material which is plasmid with me. So I am required to be duplicating it. So now you see that the bacteria in the first step, what it do is that it would duplicate all of its cellular content, not only their DNA, but the ribosomes and the plasmids as well. Now, after the cellular content have doubled, what the bacteria would do that it would move the cellular content equally to the opposite poles of the parent bacterial cell. And now since we have equally like moved them to the opposite poles, so followed by that, the cell wall and the cell membrane of this parent bacteria would split up. And as a result of that, we have formed two daughter cells or we have formed two bacterial cells and such that the genetic material which they have in them and along with that the cellular content that we have got in these two newly formed bacteria, so this is same as in the parent cell. So if you were to be writing it down in your exam, so how you could write their mechanism, you would say that at first the bacteria would duplicate its cellular components, including the cellular DNA that we have got over here, the ribosomes and the plasmids, so they would be duplicated. And then for the second one, you would say that their cellular contents, they would now move equally to the opposite poles as I have moved them here. And then in the last step, what's going to be happening is that we would be making up two cells. And these two cells, their cell membranes and their cell walls would be separated. And once they are separated, so ultimately we have made these two bacterial cells with us. So if I were to just take you to the neat diagram, here was this parent bacteria, here was its cellular circular chromosomes, we have their ribos, we have their plasmids, and then we have their ribosomes, and now we have duplicated them. After we have done their duplication, so now we have moved them into two new cells. So this entire situation or the phenomenon is the binary fission with us. Now, if I were to talk about the asexual reproduction that we have got in case of the plant, so technically it would be referred to as the vegetative propagation, and we must know that what plants actually follows them. So among them, we have got the onions, and they would asexually reproduce with the help of their bulbs. When we talk about the potatoes, so they would asexually reproduce with the help of their tubers. And for the strawberries plant that we have with us, so they would be asexually reproducing with the help of the runners, we say. Now, I would like you to be quickly looking on toward the vegetative propagation that we have got in case of the onions. So when there is growth season, so the onions, they would make some short stems. As you could see that this is a kind of a short stem that I have in case of an onion. And these onions, they would also have some remains of the leaves or maybe the entire leaves. And these leaves that we have got over here, so they would act as a storage organ. So that storage organ would mean that when the growth season has arrived, so the cells that we have got in this onion bulb, they would start to divide by the mitosis, and in doing so, they would use up the food stored in the leaves in order for their growth, and likewise, we would be making an adult onion plant. Now, if I were to quickly take you to Word, this second, you could say, an example of the vegetative propagation that we have got in the potato. So you see as that here I have shown you the top part of the plant, and here we have got the part of the plant which lies into the soil. So if you were to look into the soil, so we are forming these potatoes with us. So the situation is that during the growth season, when the top of the plant, when it receives plenty of the sunlight, so these leaves that we have with us, so they would photosynthesize. And when they would photosynthesize, so they make more glucose than would be required by their mitochondria. So the plant would convert the excessive glucose into the sucrose by combining it with the fructose. And once we have made a double sugar, or once we have made the disaccharide, which is sucrose, so it would be loaded into the phloem vessels. And the phloem vessels would bring them to these storage organs that we have got here, which are the potatoes with us. So the sucrose would be unloaded, and this potato plant has actually stored the sucrose in the form of the starch. Now you see that when the growth season would end, so the top of the plant that we have got, it would die. But these storage organs, what we are referring to them as a potato tubers, so they would keep lying under the soil. Now, some point later, when the growth season has arrived, so what this underground potato tuber is going to do, that it would develop some structures on it, and these structures they would either be referred to as buds or they would be called as the eyes. And in these buds and the eyes, the cell would start to divide by the mitosis, where the top parent plant would undergo the mitotic process to be making two daughter cells, and these two daughter cells again would give formation to other two daughter cells, and by doing so they would use up the starch that is stored here in the potato tuber, and as a result of that they would begin to develop the stems and the leaves, and ultimately we would be making an adult potato plant out of it. So now here we have got this last example to be discussing in context of the asexual reproduction or the vegetative propagation, and that are the runners with us, and this is followed by one of the frequently existing plant that we have got in our environment, and they are the strawberry plants with us. Now what happening actually is that here we have got this parent plant, and you see that this is one single plant, and out of nowhere or randomly what would happen is that their stems they would outgrow, and you see that this is outgrowing, and this would get into the soil, and over there, this mitosis of the cells would begin to happen again, and you see that new strawberry plant has started to emerge out, and after some time, they would actually be cutting their connection with the parent plant. So here you see is that this outgrowth from where this entire strawberry plant has emerged out. So this is referred to as the runner. So if you were to know the description of this process, so how you would say that there are some plants, and what they do is that they grow side shoots, and these side shoots that we have, they are technically referred to as the runners, and these runners you see on the top of them, they have got some tiny plantlets on it, and these tiny plantlets, you see that when they would get into the soil, so they would grow the roots and they would develop into a separate plant. And here you see that again, this plant that we have formed at the end, so this is genetically identical with us. So I hope that this vegetative propagation and the binary fission, that are the aspects of the asexual reproduction, it would make sense to all of you now.